Hello, Tolbert's class. This is DNA lecture number 4. DNA, lecture number 4, chapter 12. So what clues help scientists solve the structure of DNA? The clues were in Ms. Franklin's X-ray pattern that enabled Watson and Crick to take, not only to take credit, but to build their model that explained the specific structure and the properties of DNA. Chargaff's rules. Edwin Chargaff discovered that the percentages of adenine and thymine in the bases are almost equal in any sample of DNA. This is how they figured out the base pairs, and which paired with what. The same thing is true of the other two nucleotides, guanine and cytosine. The observation that A equals to T and G equals to C became known as Chargaff's rules. You should write this down. So, anytime you have so much of adenine, you're going to have that same exact amount of thymine. If you have that much of guanine, you're going to have that much of cytosine. To be more specific, if I have 10 of adenine, I'm going to have 10 of thymine equal. My total DNA has to equal 100%. So my total is 100. So far I have 20. So if I if I take 20 away from 100, I'm going to get 80. So the remaining 80 has to be split between the last two. So that would leave me with 40 of each of these in the sample. So in any particular sample, you can figure out how much adenine, thymine, guanine, and cytosine just based on the number of any one of these that you might be given. In the 1950s, a British scientist, Rosalind Franklin, used a technique called X-ray diffraction to get information about the structure of the DNA molecule. X-ray diffraction revealed an X-shaped pattern showing the strands of DNA are twisted around each other like the coils of a spring. The angle of the X-shaped pattern suggested that there were two strands in the structure. And other clues suggested that the nitrogenous bases were near the center of the DNA molecule. At the same time, these other jokers, Watson and Crick, American biologists, were also trying to understand the structure of DNA. They built three-dimensional models of the molecule. Now, why I call these guys jokers is because, like I said, Ms. Franklin, without her X-ray structure of the DNA, it's like these guys had a bunch of Legos, all separate Legos, but it matters not if you have a whole box of Legos. It matters what order you put those Legos in. She gave them, with her X-ray, the clues on what order to put the Legos in order for them to figure out how the structure of DNA was put together. Watson and Crick, too. Early in 1953, Watson was shown a copy of the X-ray pattern, stole a copy is what I was told, of these clues in the X-ray pattern, enabled Watson and Crick to build a model that explained the specific structure of the DNA, of the properties of DNA. This is the end of lesson 4. Watson and Crick's breakthrough model of DNA was a double helix in which the two strands were wound around each other. Alright, this is the end of lesson 4, and you should be able to take a quiz on Schoology over this now. Thank you.